All right, so that was the G2 checkpoint. Let's talk about the G1 checkpoint. The G1 checkpoint is probably the most crucial, most important checkpoint in the cell cycle. Remember, the G1 checkpoint is right here towards the end of the G1 phase of interphase. It's kind of a point where a cell has to make the decision: am I going to divide or am I not going to divide? If the cell receives a go-ahead signal, oftentimes from the outside of the cell, that says, yes, you need to divide, then the cell will allow the cell cycle control system to move past that checkpoint. It'll finish G1, and then it'll start synthesis, right? Remember, in S phase, this is where you duplicate your DNA. Once you start duplicating the DNA, that's the point of no return. You can't change your mind anymore as the cell. So you have to finish DNA replication and go through the rest of mitosis to divide. On the other hand, if the cell is at that G1 checkpoint and it does not receive the go-ahead signal, then it stops. It stays there, stuck at that G1 checkpoint. You like to think of it as sort of switching or looping out of the cell cycle and switching to this non-dividing state, which we call the G0 phase of the cell cycle. And you can see it here sort of looping out. In G0, the cells are not dead. They're very much alive. They're just not actively dividing. A ton of cells within your body are in that G0 state. For example, neurons. Once they are mature, they will stay that way. They will never ever go through division again. They are permanently in that G0 state, but they're definitely not dead, right? They are working tirelessly at sending electrical impulses to keep you essentially alive and functioning properly. Now, there are other kinds of cells, like fibroblasts, skin cells, for example, that might loop out into the G0 stage, but if for whatever reason they all of a sudden get the signal that says, yes, now you shall divide, they can loop back in. The G1 checkpoint is going to fall, they finish G1 and move on to synthesis and go through the rest of cell division. Okay, the last checkpoint that we have to discuss is the M checkpoint. The M checkpoint is, again, another example of an internal signal. The cell won't be able to begin anaphase until all of its chromosomes are perfectly lined up at that metaphase plate. And so you can see that in the slide here, right? The cell is here in metaphase, waiting for all of the chromosomes to line up at that metaphase plate. Once that happens, that M checkpoint will fall, and the cell will immediately go into anaphase and start separating its chromosomes, pulling them to opposite poles of the cell. You can imagine how important this checkpoint is, because if the cell prematurely goes into anaphase and all the chromosomes aren't lined up in the metaphase plate, maybe you've got some stragglers over here on this side of the cell, if you try to separate those chromosomes too soon, it might be that they don't get separated properly, and one daughter cell ends up with extra chromosomes, and the other daughter cell ends up with too few. In either case, as we'll see later in the semester, both of those conditions are highly detrimental for the cells, and likely neither one of the daughter cells will survive having too many or too few chromosomes. All right, let's talk a little bit about specifically what are the external factors that could regulate a cell maybe past that G1 checkpoint to decide whether they're going to divide or not divide. And so one of these examples of external factors are growth factors. Growth factors are molecules that are released by certain cells that will stimulate other cells, maybe nearby, maybe a little further away in a multicellular organism to go through division. The example we're going to go through in this video is platelet-derived growth factor. This stands for PDGF, stands for platelet-derived growth factor. This is a molecule, a growth factor, that's produced by platelets. If you're not familiar with platelets, they are small blood cell fragments that float around in your blood, and they're particularly important for clotting, right? So if you get an injury and you're bleeding, your body wants to stop the bleeding so you don't bleed out. And so platelets start to accumulate at the wound, sort of plugging the hole. This is just a temporary fix, though. As soon as the platelets start accumulating there, though, they start producing this platelet-derived growth factor. And this growth factor tells adjacent skin cells or epithelial cells, hey guys, you got to start dividing to fill in this injury site so we can more permanently repair the damage that has been done. So let's go through an example. In this example, we're using human connective tissue, like skin cells, for example. And so we take a sample of human connective tissue and we could, divide it up into smaller chunks using scalpels. But we still don't have a single cell sort of suspension. So what we would need to do is, on top of cutting the tissue into small pieces, we'd also have to use some enzymes to help digest away the extracellular matrix that holds cells together in the tissue. Once you do that, the enzymes chew away the extracellular matrix, then you can have a suspension of single cells within, for example, your flask here. And then what we can do is we can plate them into culture vessels. This is typically what a culture vessel will look like, where you can grow cells. You need to add media that has all the nutrients cells need to survive. You need to make sure they have oxygen and good growing conditions. So typically you'd put them in an incubator that simulates body temperature. Now, if we were to take this flask of cells and divide it between two culture vessels, in the first one, we just plate the cells, give them nutrients, give them oxygen, and these cells will survive, right? They will live inside of that culture flask, but they won't divide. They don't have that signal without the PDGF, without that growth factor. They don't have the signal that helps them get past that G1 checkpoint, and they just stay in G1. Now, on the other hand, if we were to add that growth factor, add the PDGF to the other culture vessel, again with media and oxygen and the warm temperature, those cells, not only do they survive, but they also start to divide. And they divide and divide and divide until they fill the bottom of that culture vessel with a single layer of cells, and then they stop. So this is something very interesting. Normal cells show a couple of limitations to their proliferation or to their division. They show both what is called density-dependent inhibition and anchorage dependence. So first of all, density-dependent inhibition. This means that when cells get crowded, they stop dividing. And this is what ensures that you only have a single layer of those epithelial cells at the bottom of the dish. And then even though PDGF is still present in the media, once they're crowded, they don't divide anymore. They stop. Most cells also need to be stuck to something to feel safe enough to divide. This is anchorage dependence. So typically when we plate the cells at the bottom of the culture vessel, we'll add something sticky to the bottom to help them stick to the surface of that culture flask so they feel comfortable and they can go through division. Typically we'll use something like a sticky amino acid like lysine, or maybe something as simple as gelatin. Coat that the bottom of our dish with a gelatin, and that helps the cells to stick a little bit better so that they can go through division. Now that's for normal cells. We also have cancer cells which are abnormal, right? And they disregard all normal cell cycle control mechanisms, including density-dependent inhibition and anchorage dependence, right? So they don't care, that they're crowded. They'll continue to divide, forming larger and larger tumors, or these big abnormal masses of cells. Okay, just to give you sort of an example of this density-dependent inhibition. So here's the same experiment that we did on the previous slide, where we had, our culture vessel filled with those fibroblasts or epithelial cells, and we give them media, we give them the PDGF, and they start to grow. When they grow, they fill the bottom of that flask with a single layer of cells, and then they stop. We could simulate an injury, though. We can go in there and we can, with a scalpel, remove some of the cells, much like what you would have if you got a cut, right, in your skin. Now, the interesting thing is that those cells, those fibroblasts that are on the outside edge of that hole, they're sensing that, hey, my buddy next door, he's gone. I don't know what happened to him, but he's gone. That means that I am responsible for filling in that hole next door. So those cells on the outside edge of the injury site will start to divide again until they successfully fill the injury site, and then they stop growing again. That's for normal cells. On this side of the slide, here we have cancer cells. And these guys, they don't care. So long as they have enough oxygen, and they have enough nutrients, enough fuel to divide, they will continue dividing, forming layers upon layers of cells within that culture flask.